Hello, good morning, good afternoon, good evening, everybody, wherever you are in the world. Welcome to this webinar organized by the Hyve. This is actually the first session of a series where we are going to explore innovative approaches that use the power of semantic web technologies and knowledge graphs and have the potential to transform drug safety research and monitoring. My name is Elpida Kontsiotti. I am a data modeler at the Hyve, and I'm going to be today's webinar host. So before we begin, let me run through some housekeeping. First of all, I would like to let you know that this webinar is going to be recorded. The recording will be made available after the event on our website. Also, as this is a live session, we would love to have your questions. You can post them in the questions box at the right bottom of your screen. We will try to address as many questions as possible following the main presentation of the webinar. And for those questions we won't manage to get to because of the time restriction, we will follow up with the participants via email after the session. So here's the agenda for today. Firstly, we will have a presentation by Tiffany Callahan on the topic of ontologizing health systems to facilitate translational discovery and pharmacovigilance. Then we are going to move to a panel discussion where we will explore how existing efforts in harmonizing real-world data can benefit drug safety monitoring and the potential of integrating semantic web technologies to improve how adverse drug reactions are identified using real-world data. We will have an hour in total for this webinar, and we will close the session at 4:30 Central European Time. So that's the housekeeping. Once again, thanks a lot for joining us. And without further ado, let's introduce our speaker for today. So we have the pleasure to have today with us Tiffany Callahan. So Tiffany, please join the stage. Hello, good morning.
Good morning.
So Tiffany is currently a postdoctoral researcher at IBM Research Almaden, IBM Silicon Valley Innovation Lab. Previously, Tiffany completed her PhD at the University of Colorado Anschutz Medical Campus and also had the postdoctoral position at Columbia University. Her PhD research focused on the development of realistic estimates of human disease mechanisms, and for that, she leveraged graph representation learning and neural symbolic reasoning of large-scale biomedical knowledge graphs. She's a lover of all things semantic web, an advocate of fair, transparent science, and an avid supporter of open source development. Tiffany, the floor is yours. Thank you so much.
Thank you for a great introduction. I will just temporarily be turning off my camera to save bandwidth while presenting, but we'll come back for the question and answer session. Can you see okay? Okay, here we go. So hello, my name is Tiffany Callahan, and I am currently a postdoctoral research scientist at IBM Research. I'm very honored to be speaking to you today in an area that I'm very interested in and about resources and tools that I have been developing over the last few years. I have no financial conflicts of interest with the material presented in this talk, and the work presented in this talk was primarily conducted during my PhD at the University of Colorado Anschutz Medical Campus and as part of a postdoctoral fellowship at Columbia University. This work was funded by 2 training grants awarded to me by the National Library of Medicine. And any opinions, findings, and conclusions or recommendations expressed in this talk and any of the associated materials are those of the authors and do not necessarily reflect the views of IBM Research or the National Science Foundation. An outline of my talk is provided on this slide, which will include 3 broad areas. So we'll talk about relevant background knowledge and current challenges, proposed solutions that I have been working on related to these challenges, and future opportunities within this domain. To ensure that we're all on the same page, the first part of this talk will review relevant background, which includes defining important concepts like adverse events, understanding their impact, and reviewing relevant state-of-the-art solutions in the field of systems pharmacology. An adverse event is an injury that results from medication errors, overdoses, adverse drug reactions, or allergic reactions. Adverse events are a significant public health burden, resulting in approximately 1.3 million emergency room visits and more than $3.5 billion in annual medical costs. Most adverse events are preventable and occur from known causes. Ideally, adverse events would be identified during drug development or post-marketing surveillance, but even with strict regulations, extensive experimentation, and robust reporting, accounting for every source of therapeutic and biological variance is very, very difficult. So despite decades of research, the majority of adverse events and the associated side effects are not known or really discovered until we observe them. Adverse events do not necessarily have a causal relationship to a specific drug, which makes them really difficult to identify and even more difficult to explain. Drug toxicity can largely be attributed to many sources, but primarily on-target, off-target effects, or combined on and off-target effects. And traditional pharmacodynamic modeling approaches have predominantly focused on single targets or primary endpoints. But these one-drug, one-target models are really unable to capture complex drug effects because they neglect important biological drivers of phenotypic drug response. And enumerating all potential drug outcome pairs, we believe, is vital to drug safety and will be a key mechanism for advancing precision medicine. Systems pharmacology aims to describe drug effects at the molecular level by integrating data from multiple scales across all levels of biological organization. These data are most often represented as a network or a knowledge graph, where nodes are biological entities, so things like chemical compounds, proteins, phenotypes, and edges can be thought of as relationships between these entities, so interactions, drug-target affinity. Systems pharmacology models have successfully predicted drug side effects, developed precision therapeutics, and facilitated drug repurposing. But most systems pharmacology models focus on single entities, providing an incomplete biological milieu, which can result in biased models and sometimes even implausible conclusions. So we can say that conventional systems pharmacology models potentially and most often fail to reveal important mechanisms of action and support biologically actionable hypotheses or discovery. Knowledge of a drug's phenome, or the clinical characteristics or set of all phenotypic entities expressed by a cell, tissue, organ, and organism related to a drug, is vital to identifying and understanding adverse events. Characterizing a drug's phenome requires information from many data sources, which can be difficult to integrate in the absence of formal standards. Current systems pharmacology knowledge engineering strategies lack semantic interoperability and formalized knowledge representation needed to integrate these data. But luckily, the semantic web is built on really robust standards, so things like ontologies and very expressive languages like the web ontology language, enabling more interoperable and machine-readable ways to represent knowledge. And existing knowledge engineering frameworks might be insufficient to model systems biology, which might be due to a lack of the use of semantic standards and machine-readable knowledge representation. So now that you have a better feel for the current state of things within the domain of systems pharmacology, and we all have a clear understanding of relevant concepts and the potential challenges that we need to solve within this area, I'd like to spend the next part of my talk introducing you to some tools and resources that my collaborators and I have been developing. So as mentioned in prior slides, most systems pharmacology-based analyses aim to identify drug outcome relationships using really simple networks, typically comprised of drugs, proteins, and diseases. For example, what you're seeing on the screen, lisinopril treats myocardial infarction. But these simple networks offer no explanation as to why this relationship exists. Lisinopril is an ACE inhibitor, which targets the renin-angiotensin-aldosterone system. It turns out that lisinopril treats myocardial infarction by lowering blood pressure via the reduction of peripheral vascular resistance. But there's also other ACE inhibitors which work off related molecular mechanisms, and they've been shown to cause adverse events like acute kidney injury and cardiac arrhythmia. So by understanding the full picture, in this case, using a more comprehensive representation of molecular mechanisms that underlie drug outcome pairs, we're not only able to identify important relationships, like lisinopril treats myocardial infarction, but we can also explain their mechanisms of action and identify potential adverse polypharmaceutical events which users of lisinopril may be at risk for. To try and accomplish something like this, we developed two primary projects. The first project aimed to build a robust disease-agnostic knowledge graph that was capable of representing molecular mechanisms. We called this project Phenolator. The second project aimed to build a framework that enables semantically meaningful connections between clinical concepts that have been normalized to some kind of common data model, in our case, specifically to OMOP, and concepts that are relevant outside of clinical resources, so things like ontologies. And we called this project OMOP to OVO. And our goal was to demonstrate the value of having access to both of these resources by using them to solve a rare disease subphenotyping task. Translational research requires data at multiple scales of biological organization, and researchers face significant challenges when trying to integrate these data. Knowledge graphs are used to model complex phenomena, and methods exist to automatically construct them, but they have some significant limitations, including a reliance on a fixed or limited flexibility in the way that knowledge is modeled. So once you use these types of pipelines, you're usually locked into whatever kind of system is part of the software for modeling knowledge. And a lack of relevant benchmarks to evaluate constructed knowledge graphs. So what we mean by this is once you've built a knowledge graph, there's usually not an easy way to tell how relevant or good it is without conducting some pretty large-scale experiments. So our challenge was, how do we combine the strengths of existing methods to create a more robust and ideally user-friendly system for constructing knowledge graphs? So we developed Phenolator, which stands for Phenotype Knowledge Translator, which is a semantic ecosystem for automating the fair construction of ontologically grounded knowledge graphs with fully customizable knowledge representation. This figure provides a very high-level overview of the Phenolator ecosystem, which consists of three components shown by the gray boxes that you see on your screen. So the first largest gray box is the knowledge graph construction resources. So this component consists of resources to download and process data and an algorithm to customize the construction of large-scale heterogeneous biomedical knowledge graphs. The ecosystem also contains knowledge graph benchmarks. So this component consists of pre-built knowledge graphs that can be used to systematically assess the effects of different knowledge representation decisions on downstream analyses, workflows, and learning algorithms. The final component is knowledge graph tools, and this component consists of things like cloud-based data storage, APIs, and triple stores. This figure illustrates the knowledge representation that we use to construct the human disease mechanism knowledge graph using Phenolator. The purple box represents experimental data, and the blue box contains the molecular mechanisms that we developed by integrating open biological and biomedical foundry ontologies, which are shown in green and gold boxes. Edges between the ontologies are created by integrating other data sources that are not part of an ontology, so those are the solid black lines. Dashed lines represent relationships that are inferred from the relation ontology, and dotted lines represent relationships that exist between ontologies and are imported. So the final knowledge graph we constructed was built from 12 different ontologies, 22 linked open data sets, 2 experimental data sets, and included more than 60 edge types. So these are 60 edge types we added to the existing ontologies that we imported. And this knowledge graph, again, was really designed to try to represent mechanisms of human disease. So from the central dogma all the way up to observed phenotypes and their relationships with different kinds of drugs and pathways. And this is the knowledge graph that will be referred to throughout the rest of the talk. And so this resulting graph ended up with just over 700,000 nodes and 25 million edges. And it was experimentally validated and also validated by several domain experts. Because again, our goal was how close can we get to a realistic proxy of mechanisms of disease in lieu of actually having the data to measure those mechanisms. The goal of Project 2 was to build an ecosystem that enabled us to meaningfully translate clinical concepts into a more translational space, so we could connect or map them to concepts within our knowledge graph. I like to show the image on this slide because it does a really nice job of demonstrating the value that can be obtained when converting concepts from an EHR to entities in ontologies. So on the left and right show two different ICD concepts, Fanconi anemia and acquired aplastic anemia. Below each concept is an ontological representation of the biological knowledge we have on these concepts in the form of the molecular mechanisms that underlie them. So for example, Fanconi anemia is diagnosed by an in vitro assay for DNA damage. It's caused by mutations in specific genes, which are members of the Fanconi pathway, which has the primary function of DNA repair. Fanconi anemia has many well-known phenotypes, like neutropenia, thrombocytopenia, and anemia, which are also shared with acquired aplastic anemia, which is a similar disease. So at a high level, having this information, which is not often available in large sources of clinical data or claims databases, is and can be a vital step, that we think, to improving systems pharmacology models by giving us this underlying information that isn't present alone with a concept code, but also by having more robust connections between similar diseases. Deep phenotyping, or the precise and comprehensive analysis of phenotypic abnormalities in which the individual components of the phenotype are observed and described, requires timely synthesis of multiple types of patient data. Common data models solve many challenges of standardizing electronic health record data, but are unable to semantically integrate all the different resources that we need for deep phenotyping. Open biological and biomedical ontology foundry ontologies provide computable representations of biological knowledge and enable the integration of heterogeneous data. But mapping EHR data to OBO ontologies requires not only significant manual curation, but a lot of domain expertise, both on the clinical side and on the ontology side. The goal of Project 2 was to develop a system that could generate mappings between standard OMOP vocabularies and OBO concepts. The figure on this slide provides a high-level roadmap of the system we developed called OMOP to OBO. The system obtains concepts and metadata from both OMOP and OBO ontologies and tries to obtain a mapping by first directly aligning concept codes and strings, and then leverages concept embeddings to match concepts using string labels and synonyms. Finally, if a match is unable to be obtained from the prior approaches, it can process manual mappings. Throughout this mapping process, the system tracks important metadata and collects evidence that it categorizes and outputs with the mappings. And so our goal was to leverage this system to map as large as possible a subset of condition occurrence, drug exposure ingredients, and measurement results. And before diving into the results, let me show you what some of these mappings look like. So this slide demonstrates what a condition occurrence mapping looks like. And condition occurrence concepts were mapped to two OBO ontologies, the human phenotype ontology and the Mondo disease ontology. The examples on the screen show one mapping to each of these. And note that these are very simple, high confidence mappings. So what we mean by that is there's an exact match between the clinical concept and the ontology concept. So we would call them high confidence because there's no wiggle room, they're exactly matching. Drug exposure ingredient concepts were mapped to 4 OBO ontologies. Chemicals were mapped to KEBI, organisms were mapped to the NCBI taxon, proteins were mapped to the protein ontology, and vaccines were mapped to the vaccine ontology. OMOP concepts were mapped to completeness across only those ontologies that made sense, which is nicely demonstrated by the 3 mappings shown on this slide. So again, the goal is not to create a mapping to every ontology, but to create the best possible mapping to represent the clinical concepts. So for example, the topomerate concept was also fairly simple and straightforward compared to the other 2 concepts we're showing on the screen. and it required 3 ontology concepts to map it. It's a human antineoplastic agent involving the protein interleukin-2. Finally, the most complex domain of all, measurement concepts were mapped by interpreting their results against a reference range, where the result was aligned to the human phenotype ontology, and the constituent parts, like for the drug exposure ingredients, were mapped to several ontologies depending on the concept. So we mapped chemicals to Kebi, we mapped bodily substance and organs to Uberon, proteins to the protein ontology, cells and cell-related parts to the cell ontology, and organisms to NCBI taxon. And 2 different examples are shown. The first demonstrates what a positive and negative result for a urine test for amphetamines would look like. Here we use logic on the human phenotype ontology to indicate if the result was positive or negative, and then include Uberon to indicate the substance which the lab is measured in, which is urine, and the thing that we're looking for within the urine, which is amphetamines from Kebi. The second one shows a mapping to a result that is either higher, lower, or within a reference range, and the same kind of logic was applied. We applied OMOP to OBO to all standard concepts that had been used at least once in clinical practice at a Colorado hospital and mapped them to OBO ontologies shown in the prior slides. This allowed us to build mappings for 92,367 conditions, 8,615 drug ingredients, and 10,673 measurement results to ontology concepts that represented over 9,000 diseases, over 6,000 phenotypes, 83 anatomical entities, over 2,000 organisms, over 4,000 chemicals, 132 vaccines, and 272 proteins. So a pretty robust mapping. And again, when I mentioned that we record evidence to support the mapping as we go, we also document how the concepts were mapped, which means that we have a sense of how confident we can be in the resulting mapping. We evaluated the resulting mapping in 3 different ways. First, for accuracy, where 10 domain experts were asked to manually review 20% of the most challenging manual mappings, and we found correct mappings for 73.9% of conditions, 70.7% of drug ingredients, and 92.9% of measurements. Second, the mappings were evaluated for generalizability, meaning we wanted to know how useful the mappings would be to researchers at other institutions. To evaluate them, we compared the coverage of the OMOP to OBO mappings to 24 independent hospitals, those that were part of the Odyssey concept prevalence study, and we found that 99.5% of conditions, 99.9% of drug ingredients, and 68% of measurements were used at least once in clinical practice across these institutions, and they were covered by our mappings. Finally, we evaluated the mappings on their clinical utility by comparing them to validated manual mappings when used to identify patients with rare genetic diseases using All of Us research program data. And we found that the OMOP to OBO mappings identified 99.3% of patients that were identified by the validated gold standard mappings, but we were able to do it using fewer codes and 1/3 of the query time. So using resources like OMOP to OBO and Phenolator, we examined whether it was possible to determine the genotype of pediatric sickle cell disease patients from an independent population of genotyped patients. And why does this matter? So for rare diseases like sickle cell, prevention and treatment differs based on a patient's genotype, and observational data alone are often insufficient to determine the genotype if this is not present in the patient's record. So if you don't have an indicator from newborn screen or genetic testing, it's almost impossible, not totally impossible, to accurately identify what the genotype of these patients are, given just the clinical observations we have on them. So for our study, patients with one of 4 rare diseases were obtained from a local EHR. We looked at other rare diseases besides sickle cell disease because we wanted to make this question more challenging for us, and that we wanted rare diseases that had mechanistic overlap with sickle cell disease, because we really wanted to see if having specific knowledge of mechanisms could tease out the differences between those diseases. We obtained a separate population of genotyped patient data from GEO, the Gene Expression Omnibus, and we made sure that patients were of similar age. We then built patient representations using node embeddings derived from a phenolator knowledge graph. For EHR patients, we aggregated the node embeddings for each patient's set of conditions, drug ingredients, and labs. For the gene expression patients, we aggregated the node embeddings for each gene, which were first adjusted by each patient's unique molecular signature obtained from their gene expression data. And to evaluate the patient representations and identify genotypes for each of the sickle cell disease patients, we used different patient similarity analyses. This slide demonstrates how a patient's data is mapped to the knowledge graph, in this case, a phenolator knowledge graph. First, each patient's set of conditions, drug ingredients, and measurement results are ordered by visit and concatenated into a sentence. The clinical concepts within each sentence are mapped to the entities in the knowledge graph using OMOP to OBO. Before this mapping, the entities in the knowledge graph are embedded so that each knowledge graph node embedding includes rich context from the whole graph. So for example, a knowledge graph embedding for asthma might look like the image on this slide, where the concept asthma has symptoms cough, which are caused by the mucus secretion pathway. It also has symptoms atopic dermatitis, which are caused by the gene HLA-G. And we know that albuterol and prednisone are two substances that are commonly used to treat symptoms of asthma. So the final representation for each patient contains more than just clinical codes. It contains our knowledge of known associated molecular mechanisms related to each of these codes. Before diving into the results that included the external geo patients, I'd like to start by just focusing on the results for the clinical rare disease patients from Children's Hospital of Colorado. So specifically, I'm going to show you the most important features that we identified for subphenotypes created using only clinical concepts versus when we have mapped those clinical concepts to knowledge graph embeddings. So this slide illustrates the results of the best clinical model. So on the left is when we only use clinical data, and on the right is when we have the knowledge graph derived mappings to those concepts. And this is, and on the bottom is when we've applied k-means to try to identify clusters or subphenotypes for these represented patients. So note that we found 6 phenotypes when only using clinical concepts, and 9 when we use knowledge graph derived embeddings. And what I really like to point out here is if you see on the top right, the pink or salmon colored dots are cystic fibrosis patients. And if you look at the figure below that, we're able to identify 2 pretty clean subdivisions of this group, 5 and 1, which when we discussed the concepts that represented the patients in these 2 groups, they really broke down into the 2 most well-known, and I know there are several subphenotypes of cystic fibrosis, but the 2 broadest divisions, so one based on gastrointestinal related issues and one based on lung related issues, we were able to easily split those just by using these knowledge graph embeddings. The table on the left represents the count of concepts, total and unique, and patients identified for each cluster. Due to our limited time today, I'll just focus on the results again for sickle cell disease patients, for which we identified 3 subphenotypes. And what I'm showing you here is only for the clinical data, so we're ignoring the knowledge graph-based representations for now. And I'll go through each of the most important concepts found for these 3 subphenotypes of sickle cell disease. So for the first subphenotype, we found that Lyrica was an important concept. Lyrica is a gabapentinoid, which is commonly prescribed to sickle cell disease patients to treat neuropathic pain. We also found mucopolysaccharidosis, which was found, which causes accumulation of microparticles due to the inability to break down long-chain sugar molecules. And HBSC, so that's one of the 2 primarily known genotypes or phenotypes of sickle cell disease. It's the less severe. They tend to have lower levels in their bloodstream when compared to HBSS patients, which is the more severe subphenotype of sickle cell disease. For the second subphenotype, we found sildenafil citrate or Viagra, which has been shown to prevent priapism in sickle cell disease by increasing the degradation of serum nitric oxide. And uracilidil, which is also commonly prescribed to sickle cell disease patients suffering from hepatobiliary complications like hepatic crisis, cirrhosis, and cholecystitis. Erythromycin was found for the final clinical subphenotype when using only clinical data, and it's prescribed to provide pneumococcal infection prophylaxis in sickle cell disease patients. and sickle cell disease patients are 10 times more likely to suffer from seizures than the general population, and zerontin, which was another important feature found in the subphenotype, is commonly prescribed to treat absent seizures. This slide presents the results for the sickle cell patients obtained for subphenotypes when using knowledge graph embeddings. And again, we found 3 subphenotypes, but 3 very different subphenotypes. So again, going through each of those and discussing the most important concepts. For the first subphenotype, we found diabetic autonomic neuropathy. So like sickle cell disease, it has been shown to significantly affect the autonomic nervous system, and both disorders are associated with priapism. Aphakia is characterized by the absence of an eye lens and is most commonly caused during cataract removal surgery, and cataracts are very frequently associated with sickle cell disease. For the second subphenotype, we found dabigatran, which is an anticoagulant prescribed to sickle cell disease patients experiencing venous thromboembolisms. And for the second concept for this subphenotype, sickle cell patients often experience swollen lymph nodes as a result of chronic infection and histopsychosis. Patients with sickle cell disease are at a risk of folate deficiency due to increased erythropoiesis, which can result in neural tube defects like cervical spina bifida. And there is a direct association between mandibular joint crepitus and sickle cell disease. So at a high level, what do these results really tell us? Well, if we're looking at the knowledge graph-derived patient representations, we can see that we go beyond medications as the most important concepts, which when we're focusing just on clinical concepts used to create patient representations, those are the primary features we discovered. So in other words, when we're using the knowledge graph embeddings, they're providing us more mechanistic insight, not only identifying relevant drug ingredients, but also highlighting mechanisms that exist between similar diseases. Again, diseases that are likely sharing common mechanisms or pathways. So now I'll present the results when including the sample of external sickle cell disease patients from GEO. So these are our genotyped patients. And again, our goal is to leverage information from these patients to genotype the sickle cell disease patients from our local hospital. As a reminder, the GEO patients primarily had gene expression data with little to no clinical information other than their sickle cell disease genotype and their sickle cell disease severity. Our first goal was to examine the information available in our knowledge graph for relevant genes to determine how clinically useful patient representations derived from knowledge graph embeddings to these genes would be. So the figure on the left features a t-SNE representation of differentially expressed gene embeddings, which were derived from the phenylator knowledge graph for the GEO patients, and the HBSS phenotype. And k-means clustering revealed 9 clusters, and the primary and single most significant annotations for the HBSS upregulated differentially expressed genes are labeled on this figure. These annotations were evaluated by our collaborators and were found to be very relevant for this disease. The second figure features a t-SNE plot of GEO sickle cell disease phenotypes versus control differentially expressed genes. And it visualizes the differentially expressed signatures from the sickle cell disease patients in pink and blue, where pink is the less severe genotype and HBSS is in blue as the more severe, and yellow is the control patients. And each patient's molecular signature was composed into this 2-dimensional space, which allowed us to measure distance between them. And we found that the distance between the average control patient and each sickle cell disease patient was significantly associated with a clinical measure of sickle cell disease, such that the molecular signatures of the most severe patients were the least similar to controls. These results were really important for us because they provided preliminary support for the clinical utility of patient-level representations using gene expression data. Essentially, they confirmed that representing these patients using their differentially expressed genes was a clinically meaningful way to represent them. And why this matters is that if we map the patients onto their genes in the knowledge graph, we can map our clinical patients onto the knowledge graph, and we can put both of these patients in the same space. The t-SNE plot shown on this slide represents a dimensionality-reduced view of the patient representations, colored by rare disease type, where GEO patients are shown using red and gray triangles. Patient similarity analyses confirmed that the GEO patients were significantly more similar to the EHR-based sickle cell disease patients than the other EHR-based rare disease patients. The second plot is colored by cluster assignment, which was derived using k-means. And the clustering assignment demonstrates that we were able to cleanly split the GO sickle cell disease patients into groups by genotype, so HBSC versus HBSS, and each included a similar proportion of our local EHR-based patients. So using the clustering assignment to infer the EHR-based sickle cell disease patient genotypes helped us to identify and resolve the correct genotype for 43 patients who we had discovered had diagnosis codes in our EHR indicative of both genotypes. So this analysis really demonstrated to us how our knowledge graph could be used to align independent samples of patients when important information was missing from our primary data source, and as a result, it provided us with deeper insight into our data. So now that you have a feel for the tools that we developed and one potential use case of these tools, I'd like to end my talk with some inspiration on how we envision using these resources, specifically to advance observational research-based pharmacovigilance. So while I spent most of my PhD and postdoc developing these tools, we haven't yet really gotten to apply them to pharmacovigilance, but we have a bunch of ideas on how we would now that we're here. So the first use case is to create a causal representation of adverse events. You could do something like learn a Bayesian network from informative knowledge-based priors and candidate molecular mechanisms. Ideally, one could then build models from these representations and generate causal explanations of the results. So here are some examples from our work. The first example is a mechanistic narrative for the prediction of M2 for diphlenic ATP depletion due to calcium homeostasis disruption, or M2, was predicted as one of the most likely mechanisms of toxicity for this drug. And here's what our model told us about that. So upregulated HMGB interacts with PDF-GRA, and PDF-GRA participates in positive regulation of systolic calcium ion concentration. Downregulated HMMR interacts with SLC9A1, and this gene participates in positive regulation of mitochondrial membrane permeability. Downregulated TNF-SF10 interacts with PARP1, and PARP1 participates in negative regulation of ATP biosynthetic process. So some suggested gene knockouts for further follow-up experiments at this dose and time point would be the list of genes shown here. Additionally, we can make recommendations for variants that could be related to a specific individual's response to this mechanism of toxicity, and we've output that from our knowledge graph as well. To stay on track on time, I won't go through these details, but I'm happy to share the slides. On the other side of this table, we show some examples for predictions to explain mechanistically why a specific pathway is related to certain genes. In this case, we were looking at uncharacterized but significantly associated genes for preeclampsia. And the knowledge graph is able to provide, and in this case, when we include information from the literature, justify the relationship between these inferences. One last additional example that you could use from our work, you could use a knowledge graph to learn the full universe of drug outcome combinations, identify previously unknown causal effects, and test the new adverse event hypotheses through large-scale observational database analyses. So each hypothesis of a drug outcome causal relationship could be tested by designing and executing a population-level effect estimation study across, for example, observational health databases like Odyssey. You could do comparative cohort and self-controlled case series designs to estimate the relative risk of the outcome incidents associated with each drug exposure, and even perform a meta-analysis across data sources to produce composite drug effect estimates and evaluate population heterogeneity. And of course, there are many more things that you could do. These are the things we were hoping to do but have not yet done. So just to wrap up this work, it would not have been possible without the guidance and help of everyone on this slide. And a huge thank you to my wonderful host here at the Hyve, and again for the opportunity to present and share my work with you today. And for my last slide, this provides QR codes to the associated GitHub repositories for the two major projects I discussed today. And I also include a brief list of the publications associated with each project. And with that, I would like to thank you again for attending today. And if there's any time, take any questions.
Thank you so much, Tiffany, for sharing this significant work with us. Your presentation has been very inspiring, and there are already some questions have been posted, and I would like to start with one of those, asking what approach has been used to determine K for your K-means clustering?
Great question. That's always the question people love to ask with clustering. So we tried a few different things. Ultimately, we landed on looking at different silhouettes between each possible number of Ks up to 25 and found the least amount of error associated with that distinctive set of clusters. But we also explored options where we sat down with our domain experts, in this case, clinicians who focused on rare disease, and asked them not only to interpret the data-driven set of Ks we predicted, but to look at how they might break up those patients based on their clinical attributes.
Thanks for the detailed response. Great. And I think we have time for another question. So what steps can the Odyssey community take to improve the interoperability of existing tools with biomedical ontologies? I think that's a great question too.
That is a great question. So I think that in general, I would say the Odyssey community already does a phenomenal job in working on interoperability, largely so far within the clinical space, but I know there's talk of expanding outside of that domain as well. I think the steps that would come from my expertise and just with working with these resources is to start figuring out ways that we can, the easiest thing would be start figuring out ways we can integrate these resources directly within the OMOP vocabulary and the common data model. And again, I know they're already putting in infrastructure and tooling to allow community contributions of vocabulary. So I think one step we could do as a community is start testing out what does it look like to integrate these ontologies into the OMOP common data model beyond OMOP to OBO, which is one giant step in that direction. What does it look like? What are the pros and cons? And get feedback from community members for how they would use these resources.
Fantastic. And I think that's a very nice link that can lead us to the next part of this webinar, our discussion. And for this one, I would like to introduce my colleague, Sofia Bazaku from the Hyve, who works as a data engineer in the Odyssey team. Sofia works with harmonization of diverse types of real-world data for a wide range of applications in the pharma industry and healthcare to make data integration and data analytics possible. So, Sofia, welcome, and great to have you here with us.
Hello, everyone. Very nice to be here as well, and thanks, Tiffany. That was a great talk.
Yeah, so to kick off the discussion, I would like to emphasize a key insight that came up during Tiffany's presentation, and this is the increasing availability of diverse types of biomedical data, which we didn't have access to only some years ago. And these data resources promise better evidence for improved research, healthcare, and also regulatory decision-making. And in the context of drug safety research and monitoring, they can also give us access to evidence that simply cannot become available in the tightly controlled environment of a clinical trial. But unfortunately, these data come from different data sources, are often highly distributed and heterogeneous, and this can really complicate the way we can represent them, understand them, and finally analyze them if we want to detect and monitor adverse drug effects. And for that, data harmonization becomes an essential part. And Sofia, as you've been an active Odyssey collaborator, over the past years, we've seen significant contributions from the Odyssey community in the way we leverage real-world evidence if we want to promote better health decisions and better care. So I would like you to share some insights with us into some initiatives that have already embraced the OMOP data model and other Odyssey tools to generate evidence that can support pharmacovigilance and drug safety surveillance.
Yeah, so for that, I would like to mention two initiatives. The first one would be the European Health Data Evidence Network, or for short, EDEN, with the mission to speed up real-world health research. And EDEN has helped tremendously with the adoption of the OMOP data model across Europe, with the datasets in the network including a range of different types like national registries, hospitals, or general practice electronic health records. And in addition to this large federated network, there is ongoing effort on educating people regarding the model and the open source tools, but also working together on research methods and collaborating towards open source science. And a second project that really needs mentioning is the Data Analysis and Real World Interrogation Network, or DARWIN. And this project aims to help the European Medicines Agency make informed decisions about medicines by providing timely and reliable evidence on the use, safety, and effectiveness of medicines for human use. And this is done, again, through a network of real-world healthcare databases across the European Union. And in general, in pharmacovigilance, real-world evidence from studies done by the regulatory authorities can add to the information we get from other sources like clinical trials. And the real-world evidence can support scientific evaluations and the regulatory decision-making on medicines both before they are approved and after they are on the market.
Yeah, these all sound very great initiatives with a huge potential for Europe, and let's see how it goes. The future looks very promising. And so moving on to Tiffany. Tiffany, in your research and as you highlighted during your presentation, you managed to take an extra step and build upon semantic applications to enable translational discovery using this observational healthcare data. And the tools you have introduced offer valuable biological insights and are very important when we want to analyze patient data. And one promising avenue that you already explored is the utilization of genomic data. So I would like to hear your thoughts on the opportunities of the alignment of observational health data with biomedical ontologies. If we want to develop personalized safety profiles, and if you can provide the simplified explanation of how we can leverage this approach for pharmacogenomics. So in simple words, how our individual genetic profile affects the way we respond to medications.
Great question. So I think by aligning in trusted resources like ontologies, it's possible to extend data aligned to the OMOP common data model to new resources, which can help bring in different kinds of knowledge. So when it comes to improving adverse event detection or to developing personalized safety profiles, clinical data collected from claim databases and hospitals, unfortunately, is probably not going to be enough. So not only do these resources have known biases and challenges, can be missing important populations, they really only give us one view of the underlying biology, most often the outcome phenotypes. So when things have gone wrong, by bringing in diverse sources of knowledge about diseases, compounds, and their interactions, for example, you get these additional views or perspectives of a phenotype or a problem area. And the complexity of these systems and how challenging it is to detect and predict adverse events requires diverse information, multi-scale data to span all levels of biological organization. So by making this alignment, not only does this allow us to answer a wider range of questions, but hopefully helps us to identify higher or finer precision phenotypes.
Yeah, and we know that semantic web technologies and formal knowledge representation are way more advanced and have already proven their potential in other research domains. So it's great that your research really explored the opportunities for the domain of real-world evidence. And can you share some best practices that you think can be borrowed from other research communities and can promote the translational approaches in drug safety monitoring?
Another great question. So the things that initially come to mind when thinking about best practices for me are the FAIR data principles. So for those not familiar, FAIR stands for findable, accessible, interoperable, and reproducible. And there are many good examples of how this resource has helped to ensure that data are properly documented, stored in ways they can be easily searched, and most importantly, how they aid in making research and research-related products more reproducible. There are also several tools from the Monarch initiative that are developed with this explicit goal of making translational research more interoperable and robust. So for example, some tools I commonly use, the BioRegistry. It's a tool developed by Charles Tapley Hoyt out of Harvard. It's designed to help normalize concept identifiers for hundreds of open source resources that are commonly leveraged in translational analyses. So things like Uniprot, Kemble, Kebi. I also recommend people check out BioLink and LinkML, also developed by Monarch. So BioLink is a high-level model for representing entities and their relationships. And LinkML is a tool designed to help standardize data models, which is grounded in ontologies. And while not explicitly designed for drug safety, I think these tools could definitely help make doing drug safety research easier.
Fantastic, and thanks for mentioning these initiatives. And I think these are very great and can be applied to drug safety monitoring. It's always nice to borrow best practices from other communities. So I think I've got a final question for both of you, Tiffany and Sophia. So I would like to know what you think we can realistically achieve through the synergy of Odyssey tools and biomedical ontologies when it comes to drug safety research and surveillance. And let's start with Tiffany.
I think we can achieve a lot. So I tried to hint at part of this in my future work slides. But again, just to reiterate, the Odyssey network is truly an amazing thing. The resources that have and will be developed are already demonstrating how valuable observational data can be, and if properly leveraged, how it can be used to answer really tough questions. So the value of combining external data with resources that are represented in OMOP is the potential to ask new questions and to leverage alternative forms of evidence to support what we see in the clinical data. Specifically, I think it can enable us to leverage biological knowledge of things like how do drugs work, what do drugs interact with, why do they interact this way, and that this knowledge could be used as a form of evidence, but it could also be used to derive new phenotyping strategies.
Yeah, I also believe that a combination of the OMOP CDM and the Odyssey tools with biomedical ontologies offers a great potential to enhance drug safety research and surveillance. The Odyssey community, Tiffany has said it many times already, but it has already developed so many valuable tools to offer robust infrastructure to leverage real-world data. However, moving beyond the statistical associations and distinguishing between association and causation is proven a critical challenge. Biomedical ontologies become extremely helpful and powerful when trying to understand the observed associations from biological perspective and figure out if there is causal relationship between the biological factors and specific diseases or adverse events. So by combining the Odyssey tools with biomedical ontologies, we can not only support possible drug safety issues, but also help our understanding on why these issues happen in the first place, by bringing in knowledge on underlying biological mechanisms responsible for drug adverse effects. And this integrated approach can significantly advance the way drug safety monitoring is currently performed.
Yeah, fantastic. It was really great hearing your thoughts on that and sharing your expertise with us. So I think with that, we can move to the closing of this session. And once again, I would like to thank both Tiffany and Sophia. It's been a very great presentation, Tiffany, and the opportunities that arise for drug safety monitoring but not just for drug safety monitoring, are really incredible. And for the audience, thanks a lot for joining us today. We hope that you have gained valuable knowledge and inspiration based on what Tiffany presented and also the discussion that we had. And please stay tuned for our upcoming webinars. We are having next month another session where we are going to explore how knowledge graphs can enable multi-source data analysis in pharmacovigilance. So we will hear more from us from our social media channels on how you can register for the upcoming webinar session. We look forward to reconnecting with you soon. And thanks a lot. Have a fantastic rest of your day.
Thank you.
Thank you. Bye.
Bye-bye.